So I'm Pietro. It's really a pleasure to have the opportunity to give this talk. I will discuss quite broadly the research program of my group on antibody discovery and engineering by computational design. So the reason we are interested in antibodies is that they are key tools in research and in diagnostics, and they are also an increasingly important class of biotherapeutics drugs. I have this boring slide just to establish some jargon, because this is a theoretical chemistry seminar and not everyone will be familiar with antibodies. Also, it's very multidisciplinary, so bear with me for this slide. So an antibody is, or immunoglobulin or IgG, is a Y-shaped tetrameric protein that is symmetric. It has two identical heavy chains in blue and two identical light chains. This part called Fc has to do with interaction with the immune system, and we are not very interested in it in my group, mostly because it's very well understood to a great extent. While this other part called the Fab, and in particular this red part, which are called complementarity determining region, or CDR in short, is the hypervariable part. It's the part that determines the binding to the antigen, which happens in a site called paratope. So the paratope is the binding region of the antibody. And antibody engineers over the years have established a number of antibody fragments. In particular, in my lab, we work a lot with single chain antibodies, which are just the tip of the Y, so a VH domain and a VL domain that retain monovalent antigen binding, and with nanobodies, which are just the VH domain, but they are able to fold and function independently of a VL domain, and they are typically called VHH. And these were originally isolated from camelids, but they are important in research and biomedicine. And again, these are the three binding loops of a nanobody, 1, 2, 3, and a full-length antibody would have 6 binding loops. But the most important one and the most variable one is CDR3, which I will be mentioning throughout my talk. So with this jargon out of the way, if you want to make a new antibodies for a target of interest, nowadays you can use two classes of technologies that are very well established and that have been very successful. Either in vivo discovery, so immunization of animals or isolation from patients, or in vitro discovery, where you build a large combinatorial DNA library and then you screen it for binding with things like phage display, which was the Nobel Prize award of Greg Winter here in Cambridge, but also other display techniques. And again, these have been very successful, but they remain quite time-consuming and costly, in particular the in vivo route. And more importantly, there are a lot of unsolved challenges. In particular, I mentioned 3 for simplicity. It's very hard to use this technology to target a specific epitope, so a specific region of your protein that may be functionally relevant. It's easy to get binders to your protein, but where they bind is very hard to determine. It's also there are also some classes of hard targets, like disease-related protein aggregates or membrane receptor of proteins that are part of highly homologous families, and it's very hard to use this technology for what I call multi-trait optimization, or in other words, this may be quite good at give you good binders, but it's not guaranteed that these binders will also have high stability, high solubility, low immunogenicity, and all these other traits that are needed to develop an antibody into a product. So this space here is really the gap we are trying to fill with our work. And the way we are trying to fill it is by introducing a new generation of technologies of computational design. And the reason we think this is possible is that we now have access to, of course, powerful computers and powerful AI and machine learning, but more importantly, I believe we have a lot of high-quality structural data and really a lot of high-quality sequencing data from NGS and deep sequencing that was established here by Shankar and David Kleinerman. And then we have the ability to do highly quantitative biophysical measurements, and I think that taken together, this paradigm shift will enable the development of the third generation of antibody discovery method, exactly like it happened in the past for a first generation of in vivo methods and the second generation of in vitro methods. So I'd like to, at least try in the time I have, to share 3 stories with you. One about our computational design of nanobodies, in this case, targeted predetermined epitopes. Then quite a story on predicting nativeness and humanization, which will become clear as I walk through. And finally, a story on multi-trait optimization. So one of my earlier contributions to the field, and this is probably the only slide I have that contains work from when I was a PhD student or a postdoc, was about targeting proteins involved in misfolding diseases, so amyloidogenic antigens. These are diseases like Parkinson or Alzheimer's that are characterized by the formation of these long and highly structured amyloid fibrils. And so we needed a way at the time to design antibodies for specific epitopes in this protein. And when we did this for a protein called synuclein, which is an hallmark of Parkinson's disease, when we designed one loop, we could get decent binding affinities, but nothing too outstanding in the micromolar range, while when we design 2 loops, which is what natural antibody would do, would bind with at least 2 of these CDR loops, we would get affinities that are in the ballpark comparable with those that you get from in vivo or in vitro techniques. We have also been able, in collaboration, to give these antibodies, or nanobodies, I should say, to C. elegans models of protein overexpression. So what happens here is that as these worms age, they overexpress human synuclein, which aggregates, so they get all paralyzed, essentially, but they do not get paralyzed when you treat them with an anti-synuclein nanobodies that prevents the aggregation. And slightly more recently, we have also been able to go after a traditionally hard target, which are these oligomeric aggregates that are intermediate that force between on the aggregation pathway. So at the end, you see this long structure amyloid fibrils. Everything starts from a functional monomeric human proteins, and in between you populate these fleeting intermediates that are called oligomers and that are reported to be the most neurotoxic species. But they are a very hard target because they are quite short-lived. Either they convert in fibril or they dissociate back, and they are also very heterogeneous. And what we were able to do with some design and a lot of experiment was to isolate nanobodies that preferentially bind to oligomers a lot more than they bind to monomer or fibril. And this approach was quite successful, so it spearheaded a number of collaborations, and I would say that we validated it well beyond the sort of proof of concept. Now, this is work from more recent year, where essentially we develop a much more general approach that now is not limited to amyloidogenic epitopes, but can be used to target structured epitopes, whose surfaces of a protein whose structure is known or predicted by AlphaFold, for example. The video that played was the coronavirus spike protein that I don't think needs any introduction. And the red part of the spike protein is the part that binds the human cells and mediates the entry. So it's a well-known neutralizing epitope. What we have done was to design a CDR loop in blue to bind on this epitope, and then we grafted it into a nanobody. And we saw that this nanobody bound with KD in the high nanomolar range, which is quite remarkable for de novo design, but still not quite as good for therapeutic purposes. In gray, we have negative controls, which are important. So this would be the exact same nanobody, but with a different CDR3 loop that doesn't bind. We have conferred the binding in solution as well, again with a negative control. And finally, we have seen that the binding of the nanobody at least partly inhibits the binding of the human ACE2 receptor, as you would expect for a nanobody that binds where we designed this one to bind. So it's a sort of indirect confirmation of the epitope. We have done a couple more COVID nanobody in this work, and we have also done some for this protein in gray, which is human serum albumin. And again, for these 3, we found a very similar story. So they still bind similar KDs in the high nanomolar range. We can confer the binding on the immobilized sensor as well. And in this case, we can also do a binding competition experiment. This is a bit messy, but it's quite intuitive. So we designed these 2 for essentially the same epitope. So we expect these 2 nanobodies to compete with each other for binding. while this one is designed to bind on the opposite side, so it shouldn't compete. And indeed, this is what we observe here. The blue and red trace are similar to the positive control for competition, which are the nanobody competing with itself, while the dark red and dark blue traces are similar to the negative control for competition, which is buffer, so nothing, competing with the nanobody alone. And this pattern is exactly what we would expect from the design, which sort of confirm that we are hitting the places we designed to bind to. And then we quickly ask also the question of stability for this design. This y-axis is a proxy for stability, is a melting temperature, so thermostability. And we find that our designs, which are the 6 one we did in the paper, I only showed you 4 in the presentation, are in the ballpark in the higher end of the distribution of natural nanobodies. So to conclude on this first story, I guess we have shown that the de novo design of antibodies that target predetermined epitopes, at least some predetermined epitope is possible, and some is because we use a fragment-based design procedure that is a bit limited by what's available in its fragment database. These resulting KD are very good for de novo design, and I should also state that in this approach we designed this nanobody and tested them in the lab as they were, while a lot of other people who have designed binding proteins or binding antibodies typically design a library, like hundreds or thousands, and then they screen it for binder. But still, this KD is far from therapeutically relevant ranges, so we need to get 2 orders of magnitude better. We have negative control that demonstrates selective binding, which is good, but the actual specificity is likely not sufficient for diagnostic application. And finally, these nanobody have rather artificial sequences because of the way we design the interaction, so they're likely immunogenic. And these are areas for improvement. And if you want, these are the challenges of computational design of nanobody we are trying to address at the moment in my group. And now I want to take a step back and briefly talk about the challenges of antibody development much more broadly. So now it doesn't matter if you computationally design it or what, you will be faced for these challenges, especially for therapeutic application. So on the functionality side, you have challenges such as binding to a functionally relevant epitope, like the neutralizing nanobody example. You have challenges for species cross-reactivity, which is often a requirement for clinical trial because you need to get tox data in animal models. But perhaps most importantly, the main challenge is that of obtaining the desired biological activity at a relatively low dose and administration frequency that make it feasible for patients. And this means that the binding affinity of therapeutic antibody is typically in the tail of the distribution of the binding affinity of natural antibodies. So you need picomolar or single-digit nanomolar. And then on the developability front, we are asking our products, whether for diagnostic or therapeutic, to survive a lot of stresses that are related to manufacturing, formulation, shipping, and administration that these antibodies have not evolved to endure. And developability is an important concept, is an umbrella term that is defined, if you haven't met it before, as the likelihood of a drug candidate that has suitable functionality to be developed into a product that can be formulated to high concentration, retain a long shelf life, and be safe to use in patients. And if we just look at some number to get a sense, for subcutaneous injection, we need concentration of a single antibody up to 200 milligram per ml, while the total IgG concentration of all antibodies in our body is typically an order of magnitude below. And also the half-life of antibodies that they have evolved with is roughly 20 days, but we need products that are stable for at least 1 year. And this leads to challenging such as aggregation, unfolding, viscosity, and chemical degradation. So the bottom line of this slide, which was very crowded, is really that to develop antibodies, often one needs to push all biophysical properties beyond the typical natural level. So that's the key challenge, really. And what are these biophysical properties? This is a sort of example, but it's really quite a lot. Some of them underpin functionality like affinity, specificity, interaction with the immune system, or fancier modalities like pH-dependent binding or multispecificity. And some of them underpin developability, like stability, which is the population of the unfolded state versus the folded state, or colloidal stability, which is the long-term integrity of a formulation versus aggregation, solubility as a critical concentration, expression yield, viscosity when you inject, and pharmacokinetics. And these are not only are a lot of properties, but they are also closely intertwined, in the sense that if I make a mutation that, say, improves binding affinity, it will impact a lot of the other properties, and most often in a negative way. So the metaphor I like to use is that of the rubric cube, where it's relatively easy to solve one phase of the cube. So you can use phage display nowadays quite reliably to push the binding affinity beyond the natural levels, but this usually comes at a cost over the other properties. So it's much harder to solve the puzzle. And again, the bottom line, and this is well known in antibody discovery, is that the hits with the best functionality are often not those with the best developability profile, and vice versa. And the way this problem is currently solved is by looking for the needle in the haystack. So people in pharma company and so on will screen more and more until they find the right one. If they don't find it, they close the program, they move on to the next program. More or less, that's what happens. In my group, what we are trying to do is to develop a toolkit for computational antibody design, or in other words, a number of computational methods that can deal with this complexity and that we can use in tandem to get the right balance. So this slide is very crowded. I won't have time to present all of these, but very briefly, in the blue shade, there are our algorithms to design binding. I already discussed this story where we use this fragment-based design to get this high nanomolar KD. We are now using a lot more AI, but we still don't have enough solid data for a story here. In the pink shading, we have a number of tools that I call for hit selection, so things that can rapidly, from the knowledge of the sequence alone, predict the various properties that underpin developability. So in these panels, the x-axis is always our prediction from the sequence alone, and the y-axis is various experiments, in this case of stickiness or propensity to self-assemble or be polyreactive, of solubility. This one would be stability, so the measured melting temperature. And the bottom line is really that our prediction can almost replace experiments. And this is a generalization of these approaches of predicting solubility. Now the measurement are the green dot, and the prediction is the continuous blue line, and what we are looking at is solubility as a function of pH, which is an important parameter for formulation. This story of nativeness I will discuss more in depth now. And finally, in the yellow shading, we have what I call methods of lead optimization. So here you would take an antibody as an input, maybe something we design, maybe something that came from phage display or immunization, it doesn't matter. And these methods would predict mutations automatically that improve one or more traits, and I will also discuss these in more details. Let me take a look at the time. Okay. So the next story I want to share with you starts with a consideration. And the consideration is that the discovery from animal immune system or from patients remains today the gold standard for therapeutic antibody development. So these are all data that show that only 15% of the clinically approved antibody come from in vitro, and actually this percent has become even lower since, because a lot of these animal models have now been introduced with a fully human immune system and things like that. And this is surprising at first, because if you have read any of, I don't know, Greg Winter Nobel Prize associated reviews, you may have read that these in vitro methods offer a lot of advantages. They are faster, they are cheaper, they offer much more easy antigen presentation. The antigen can be non-immunogenic, it still works, so it's surprising. And the reason in vivo approach still dominates is that of what I call in vivo properties, or in a simple line is that immune system derived antibodies typically have a good balance of in vivo properties, and this includes specificity, half-life in vivo, and safety and immunogenicity. And these properties are incredibly hard to measure experimentally and in good throughput, which makes them incredibly hard to predict. And again, this is a statement from a company presentation. It is reported that roughly 40% of the biotherapeutic in the market have their activity reduced by some anti-drug antibodies that our bodies make against them. So this is an important problem because it's telling us that if we want to do computational design, we should be able to predict these properties, except is impossible because they cannot even be measured reliably. And so what we wanted to do instead was to bypass the challenge of predicting them directly to instead copy the immune system, or in other words, achieving a good balance by designing antibodies that are indistinguishable from immune system-derived one. And the reason we can do this is that we have a lot of sequences from immune system-derived antibodies that we can exploit with AI. So this thing is called a vector quantized variational autoencoder, but it's late in the afternoon, so I'll just tell you how it works. So the way it works is that you give it a lot of sequences, and it learns a latent distribution of where these native sequences exist. Because it has an information bottleneck and also some other things. And then when you use it, so after you have trained it, you give it the sequences you are interested in, and what it does is essentially estimates the distance from the learned distribution. So if the distance is low, we say that you have a high nativeness. So your sequence is very similar to the one of immune system derived. If the distance is high, then you have low nativeness. So this is what you need to know. Then we train it on human antibody sequences, and then this is a test set, so some human antibody sequences we didn't use for training, and we see that the human one are predicted with high nativeness or humanness in this case, while the mouse one that is never seen before are different in some way, and so are predicted with low humanness. So we can use this already for hit classification and ranking. We have done it also in more sophisticated way. So this is the same thing, just more quantitative with area under the curve and so on. For monkeys in green, mouse again. These blue ones are interesting. So these are artificial sequences we make in silico. This is a control that few people do in the field of machine learning and AI, but I think they're very important. So what we have done here is that we have made the fake sequences using the human amino acid distribution. So these would be essentially fully human sequences, except for the fact that each position in this sequence has been filled independently of the other, because we have done it randomly. And so the methods has learned that these are different somehow. It separates them out very clearly, which tell us we have learned some higher order relationship between amino acids. So we have not just learned the frequency of amino acid, but also how they go together, which is important for design. And finally, we made it interpretable. So together with the score that we plot here in this x-axis, which is a score for the whole sequence, we have a single residue resolution profile that tell us the humanness at the regional level, so we can use it to design mutation. This example in particular is a mouse antibody that has been humanized experimentally and became an approved therapeutic. And you can see that the humanized variant in purple has no regions of low humanness, except in the CDR region, which are here, because these are still the mouse sequences. That's how antibodies have been humanized historically. You take the mouse CDR, you put them in a human framework, you hope that it works. And so what can we use this method for? And one way is to do nanobody humanization, because nanobodies are becoming increasingly important, and we had the opportunity to do this. So on the x-axis is the nanobody nativeness, and on the y-axis is our humanness. So these are both predicted by our method. We have various sampling pipeline to suggest mutation automatically, and we make this design. So the requirement we had was that the nanobody nativeness was not lost upon humanization. And we compare with another method that came out as we were developing our own, that is a rather sophisticated structure and frequency based. And the other method we can already see is very good at improving humanness, but this comes at the cost of nativeness. And when we move them to the lab, we see that this also, let's focus just on the bar plot, this also comes at the cost in binding KD. Indeed, this one doesn't even bind. This is a very tight binder that was optimized by multiple cycles of in vitro evolution after lama immunization. While in our case, at least the one of the red, and so essentially the one where we don't change buried amino acids, which you shouldn't change really, we retain the binding at par or maybe even slightly better than the wild type. And when we look at stability, we also see where we tend to improve or at least retain stability, while other methods have a loss here or they have just retained it. So that's was encouraging. So we use this approach in partnership with a company that does CAR T cells. So a CAR T cell is a T cell that has been engineered to display, in this case, VHHs on the surface, so it can recognize tumor-associated antigen, in this case, this antigen here associated with glioblastoma, and when the recognition happens, the T cell will kill the tumor cells. So initially, this company had an alpaca-derived nanobody that was binding the target very well. We humanize it, we retain the binding in vitro, no surprises. And then we went to the more ex vivo contest. So now we are on the surface of the T cell with our nanobody, and we are seeing if it can kill cancer cells. And the answer is that it can. So all of them, so the wild type and our two humanized version, can kill essentially all of the cancer cell ex vivo, unlike untransduced T cell. So the same T cell without the CAR, essentially, as you would expect. And things became more interesting when we look at off-target binding. So here is killing cells that don't have the antigen. And here we can see that the wild-type CAR with the nanobody kills quite a lot of these cells. And this shouldn't, right? This is a toxicity flag, even at relatively low T cell to tumor cell concentration, while the one we optimized are doing better, especially the enhanced one is doing much better. And when they took it to in vivo, we see that in the NT, so the T cells without the CAR, the tumor grows in 14 days in all the mice in the arm, while in the R that had the CAR and the armor CAR, but this armor part is a contribution for the company, not from our lab, the tumor was cleared after 14 days in all the animals. So to quickly wrap this up, we have shown where we can design antibodies that are undistinguishable from immune system derived one, and we can use this method for screening existing sequences or for humanizing available nanobodies, and indeed not just humanize them, but perhaps also improve their in vivo properties, including specificity. And now, with whatever time I have left, yes, I have left some time, I want to switch gear and tell you a bit of this other story on the optimization of multi-trait optimization of stability and solubility. So why stability and solubility in particular among all the other properties? Because these two properties are quite important for developability, perhaps the most important, because together they determine aggregation propensity, degradation, especially stability by proteases, and of course denaturation. And together they've been shown to determine colloidal stability. They correlate with expression yield, which is very important if you want to manufacture a product. They often anti-correlate with viscosity. High solubility correlates with low polyreactivity. Polyreactivity is stickiness, let's say in vivo, or poor specificity. And high stability correlates with long in vivo half-life. This has been shown over and over. So together they typically embody better pharmacokinetics, and also they are quite crucial for new things like oral delivery or inhalation delivery, things that are not established but people are working on, as well as new formulation modalities. So this is the rubric cube challenge I introduced before, because it's well known that mutations that improve solubility often worsen stability and vice versa. So the way we do it, quite briefly, is that we run an algorithm called CAMSOL that we developed back in the days to identify solubility liabilities. And then, if this works, yes, we also identify other conservation liability by looking at a lot of other nanobody or antibody sequences. So this would just be an alignment of a lot of sequences. Actually, a representation of the position-specific scoring matrix, where the top is the most probable sequence and the one in orange are essentially residues that are never observed at that position. And then we singly model our mutation, and we predict with an energy function the effect on stability, and with our method the effect on solubility, and finally we combine them together to make design where we also check for synergistic effect of mutation. And the point I want to make is that this is very important, this part where we look at similar sequences, because it grounds the algorithm in a space that is evolutionarily and biophysically grounded. And in fact, we find that by doing so, by restricting the space, we substantially lower the false discovery rate of this energy function. So when we just look at the space of amino acids that are more probable than the wild type and are enriched, we have almost a 10%, or if not more than 10% drop in false discovery rate, which is important for design, because if you make false positive prediction, you would take them to the lab, and you don't want to do that. So we took this to the lab. This in particular is one nanobody. And these are now experimental data. And we can see that we improve relative solubility for all the variants, as well as stability for all the variants. The wild type is here. This is another readout of cross-reactivity / solubility. And again, the improvement is there except for mutation to positive charges, because these become a little bit more sticky. And this is well known in the literature. And this improvement, just to put it in context, is very substantial. So the stability has improved by almost 14 degrees, which is a lot, and this retention time in this assay by 44 seconds, which is basically the whole dynamic range of this assay. And then they all retained binding to the antigen, apart to the wild type, except the negative control, of course. So this got Novo Nordisk excited, so we collaborated with them, and now instead of doing a single nanobody in my lab, we did 2 more nanobodies and 3 single chains, so full antibody, 2 of which are clinically approved nanobody. Adalimumab is also called Humira, is the best-selling drug of all time, so in other words, it's a very optimized antibodies, and in fact, when we look at solubility, we couldn't really improve it anymore, but we could improve stability. So actually, it's easier to look at the things that didn't work, which is only 2 mutations. This one here, we thought it would make things better, but didn't. And this one here, which is the same mutation present in 2 design, which also we thought would have made things better, but didn't. But overall, it was a very good success rate. And very recently, we also applied it to enzymes, so not antibodies, but some of these methods are generally applicable. This is an alpha amylase, so an enzyme of high industrial relevance, and is an hyperstable one because it comes from a thermophilic organism. So we did a few design, we did a few design, and we showed that we retain the hyperstability. We cannot improve it anymore, but it's already super stable. We retain, perhaps improve, but I guess retain is fair, the activity, and we improve massively the solubility. Again, this improvement of more than 9% is quite a lot in this particular assay. So to wrap up this other story of multi-trait optimization, I guess we validated this with 1 enzyme and then 6 antibody fragments. We find that we can accurately design single or multiple mutation. We find a very high success rate, or a very low failure rate, if you want. And again, I didn't show this data, but only 2 of 33 substitutions we did in the antibodies affected the binding to the antigen. So again, this is critically multi-trait optimization. You don't want to improve solubility and stability by changing affinity. So we put quite a lot of work into making our methods as available as possible to the community. This is an effort that I started years ago, but I've done it even more in my group. So we make them available for download and as a web server. With the web server, users have to register. So some of them, we don't know where they live because they have used emails like Gmail or whatever. But 2/3 of them have used email like at chemet.ac.uk, and then I know where they work. And this is where they work. So it was quite nice to see this. I'd like to thank my group over the year, former mentor and collaborators, a lot of industrial collaborators, the people who gave me the money, and I guess I'll leave you with a visual summary of the 3 stories I shared with you.